Okay, for the sex-linked genetics, you need to understand that you are looking at the male versus the female. And so we use the alleles that represent the male and female, which are XX for female and XY for male. So if you notice here on this Punnett square, the female, the mother is at the top. She is an XX, and the male is at the left side. He is the XY. And this little letter is the hemophilia trait. And if you notice, the mom in this example is a carrier. One of her X's has the little H on it. If both of her X's had the little H on it, she would be an affected female. But she has she is a carrier. And on the left, the male is affected with hemophilia because this is an X sex-linked trait. And he only has one X because he is a male. And because his X is affected, he automatically gets the disease. So if we multiply the alleles by each other to find out what the possible offspring could be, you'll see that this male's X that's affected times this female that is not affected gives you this in the Punnett square, which means it's a female that is not affected but is a carrier because one of her X's has that little H. And what that means is that some of her eggs have the carrier gene in it and can be passed on to her offspring. In the second cross over here, which is an affected female with a carrier X on the female, affected male, and a carrier X on the female, that female is affected. Both of her X's have the trait, which means she will have the disease. Over here, you have a Y from the male being crossed with an X from the female. We put these in alphabetical order. And if you notice, this male is not affected. And that's because this is an X trait. And this X up here did not have the trait on it. So when it mixed with this Y to make this boy child, that boy child is healthy. But in this case, where the mother's carrier X that had the trait crossed with that Y, then you have a boy who automatically has the disease. Now, he automatically has it because he only has one X. And if that X is affected, he automatically gets the disease. That's very important to note. Over here, we have something called codominance. Codominance is something that you see a lot in animals. This is where we get the black and white striping of zebras, for example. This is where we get the black and white dots on Dalmatians, for example. So what you see is two traits showing up in the same ratio. So this example is of a white chicken being crossed with a black chicken. And if you notice, we use different types for these particular crosses because this is codominant. So we don't use the same letters that we did back here in the Mendelian genetics, where we use the same letters where the dominant and a recessive trait. In codominance, both traits are dominant. So on the white, we have WW, and on the black, we have BB. So when they mate and they form an offspring that is speckled, the trait becomes BW. They're seeing the black and the white in that third chicken. Here's another example with flowers. You have a red dominant flower mixed with a white recessive flower, and you end up with a red and white flower. And we're going to talk about the pink flower in just a moment, but you need to know that when you mix a dominant with a recessive and you get half and half showing up in the offspring, that is a codominance genetics example. In the incomplete dominance, where we get the pink flower, that is a completely new and different trait that is observed. It is a third trait, and it is what you would consider a blending of the genes and the traits. So you have the red and the white make a pink, a purple and a white make a lighter purple, etc. So you would see actually a third brand new trait. This is how flowers have become different colors over the years, and how roses, for example, have become different flower colors, is from genetics and from the people who raise those flowers, mixing them on purpose so they get these beautiful colors. So that should help you when you're going back into your pedigree to look at your traits that you're looking at. You only need to trace one trait. I know there was a student who was trying to get all sorts of different traits in there, and that is not necessary. You would need a separate pedigree for each one. I also would like to remind you that you will be doing a Create a Kid lab. So this needs to be printed out. And when you're done with this lab, each parent, so each one of you, you'll be doing this in partners, will draw the child that you create on a typing paper without looking at the other parent's drawing. So you might want to go to separate sides of the room so you don't cheat. And at the very end, you will show those to each other to see how they compare. These are not going to be pretty kids. I can promise you that. It's a lot of fun. This is the Create a Baby Lab. I will post this in the classroom, but it does need to be printed out because you have to print out write on page 2 your results in order to draw your kid. And then page 3 and page 4 actually show you what those traits will be. So you're going to be flipping pennies. I believe head equals dominant, heads tails equals dominant recessive, and tail means recessive. And you will be filling out what these kids are going to look like. There is some down here where we talk about the codominance and incomplete dominance. That will happen. There are 2 traits that you will be doing that on, and that is on your worksheet. So please read that carefully and ask your sub for help if you need it. It will also help you to do these vocabulary words if you haven't done them already. So I will post that in the classroom with this recording. Last but not least, I would like to remind you that Friday is a half day, and so plan accordingly. And also we have Beat Back Bufflegrass Day and the Swan Wash Project this Saturday from 7 until noon. I will be there. I will be handling the registration and dividing up the groups and filling out the liability forms. So come out on Saturday from 7 to noon to help out. We really could use your help. Thank you very much and have a great week.